Hello, my friends. Thank you so much for joining me. Today is part 4 of this biotechnology and biological pharmaceutical products series. And what I want to focus this presentation on is the quality by design approach. And I want to specifically think about the manufacture of monoclonal antibodies and how quality by design can be used to improve and maximize clinical performance and potentially reduce adverse drug reactions. So this presentation, hopefully it won't be too long, will be an overview of the development of monoclonal antibodies and how we can engineer not only the antibodies themselves, but the cell substrates that are used to produce them to maximize efficiency and minimize adverse reactions, therefore optimize optimize manufacture. So I'm going to also give an overview of the manufacturing process in the context of quality by design and perhaps where we are moving forward. So the first part is the design of the molecule. This is the biological entity and the specific quality attributes within that molecule itself that enables an improved or enhanced clinical performance and reducing the clinical adverse drug reactions, and also to optimize manufacture. So where do we begin? How do we design a molecule? In this case, it's a biological entity or a biotech product. It is a humanized monoclonal antibody. What quality attributes should it possess? Well, we start off with the target product profile, and this is a prospective assessment, if you like, where we are taking into consideration the disease, the disorder, the condition that the molecule is going to treat. We look at the clinical aspects and we look at the drug product aspects. For the clinical aspects, we're looking at the efficacy, we're looking at the potential mechanisms of actions or mode of actions, we're looking at the potential side effects and safety issues that we could be confronted with based upon prior knowledge, prior experience and platform knowledge. And also for the drug product aspects, we're looking at, for example, compatibility. We're looking to see whether or not this drug product that we're going to design, how will it be applied, how would it be administered, what would its safety or its compatibility be with the solvents that are used in the clinic or in a healthcare center to deliver the products. What's the shelf life? How is this going to be presented? What's the storage conditions? How does this look? So these are the things that we consider, and we start off prospectively with a target product profile and we build upon it with time. The target product profile could be a target development product, or it could be kind of like a phenotypical development program, where in the target development program you're looking at, say for example, a target protein, a protein or some kind of receptor that the antibody might be binding with and producing its therapeutic effect in that way. But for phenotypical, you may also be binding your antibody to a target protein, but you're looking to address perhaps the symptoms and not necessarily the actual biochemistry of the actual disease. So the quality attributes that are necessary, or the properties or characteristics of the molecule that are necessary for this intended use, you can gain those an understanding from that, from the platform knowledge that you have, from the scientific knowledge that you have, and you can design or you can include design features related to your mechanism of action and design the actual molecule itself to minimize ADR, adverse drug reaction, and to maximize your clinical performance in use. And so what I find really interesting is how that we've got to the state now where we design a monoclonal antibody, and we put a lot of input and science and adaptation and engineering into the actual genetic sequence and into the actual cell substrates that are used to manufacture and produce these antibodies. So I just put together this very simplistic diagram, if you like, of the antibody, because I think it's always good to know what we're talking about. And I just wanted to quickly run through them. But, you know, antibodies are part of the body's adaptive immune system. They're produced by the B cells, and they are very selective. They're highly specific to their targets. And they can be, well, the targets are usually the epitopes on the antigens, such as viruses or cancer cells, or cytokines or cytokine receptors. So they can be used therapeutically, diagnostically, but they can also be used for other things like, for example, immunological assays, for ELISA analytical procedures, etc. So they really are versatile biological entities. They have key regions that carry out certain functions, and this is why I've got this diagram here. So we have like a heavy chain, which is the which has a variable region and it has a constant region, and then it also has a light chain. But this is the heavy chain, which I've identified here because I think it can be quite complex. And then, in the next slide, so this is the light chain, showing the variable regions on the light chain and the constant regions on the light chain, and the hinge and the disulfide links. And as I said, the antibodies are highly selective and highly specific, and actually the variable regions are the part of the fragment that contain the complementary determining regions, and the complementary determining regions are there's three on the heavy chain and there's three on the light chain, and they contain these complementary determining regions, the CDR. They contain or form the paratopes, and those paratopes are what interacts with the epitopes on the antigen. I'll show this in another slide, but the constant region, this is what is responsible for the effector functions of the antibody, and that's the antibody dependent cellular cytotoxicity and complement dependent cytotoxicity. I'll show those on another slide. So I just want to talk about how we have different forms of antibodies. So, once you've got your amino acid sequence or your genetic sequence for your antibody, there are various forms that can be produced in cells, and these are the fragmented antibodies, which I've already discussed. You can also produce chimeric antibodies. This is where we graft the variable region of an antibody onto a second antibody. So if we want to graft the variable region onto a human antibody, then we have a humanized chimeric antibody. And then you have fusion, antibody fusions, where we fuse variable heavy chains or single variable fragments onto an antibody. You can also combine antibodies to produce two antibodies where we have bispecific or trispecific, where you have 3 antibodies, or multi-specific antibodies, and this is using protein engineering techniques. So these different forms are determined by genetic engineering, and once a sequence has been determined by next generation sequencing, you can then produce these types of antibodies from specific or specified cell substrates. So, as I said, this structure of this antibody, I'm just going to use this to represent IgG1 monoclonal antibody, and to discuss the structure and how changes in this structure can, or design in this structure, can impact the efficacy and safety and produce an optimal effect. So, you know, I guess you could consider it to be a quality by design approach, where you look to build in the attributes and characteristics into the actual antibody in order to maximize clinical performance, and also in order to maximize the manufacture. So from prior knowledge, we have an understanding about the antibody structure. We know that changes in efficacy occur because of, say, for example, the glycosylation patterns. You know, human glycosylation patterns are, you know, quite important. They impact on efficacy, or they can, if you don't have the right glycosylation patterns, you can end up with adverse drug reactions, or where the body potentially recognizes the monoclonal antibody as non-self and basically instigates an immune response. Or even if you don't have an adverse drug reaction, the body can just simply remove the monoclonal antibody from its system. So glycosylation, which is post-translational modification, it's less well controlled than, for example, genetic sequences of your amino acids, you know, where you form your transgene. But we can develop a means of engineering the host cell and the transgene, you know, to get the glycosylation that we want in order to promote our desired therapeutic effect. So we can say, for example, produce amino acid residues that promote the required glycosylation, and this could be through the introduction of various mutations that could cause that effect. And that's something obviously that a company with that kind of information, the company would hold in their confidential information. Those disulfide links that I've got in here, they're really important because they are known to break or shift, and when they do, we call these disulfide shifts, that can cause changes in the overall structure. So they're really important for the conformation of the antibody. And what we have done, or what I've been able to locate, is that we have engineered the monoclonal antibody with amino acid residues that can perhaps stabilize those disulfide shifts and prevent the shifts from occurring, and have a much more stable and effective drug product. So that's kind of like an example of where we have designed the molecule to provide the maximum clinical performance and minimize you know, adverse drug reactions. Disulfide shifts usually occurs on stability. You see them in the SDS page, and you see it in isoelectric focusing, and you also see it when you get aggregation. So when you get aggregation as a result of the fact that the structures have changed, and this can potentially cause issues. Deamidation is something I just mentioned briefly there, and that's important. You want to avoid that, specifically along the fragment antigen binding regions, because that's obviously what's responsible for binding to the target protein. And if you have deamidation, then you're going to change that, and you can therefore engineer your antibody with residues that do not lead to deamidation, or at least leads to it less of the time. And therefore, if you think about it, you can, by taking these approaches, you can produce antibodies that are designed to have a higher half-life in vivo, in the body. I don't know if there's anything else I should say about this.